Hi, everybody. Today we're presenting occurrences of epiphytic algae on red algae. My name's Tito and my partner's name is Ivan. Our study focuses on red algae in the genre Kappaphycus and Leukemia in Malaysia. Uh, so algae is a major agricultural crop and an integral part of the economy. Uh, and red algae was introduced in the 1970s for the purpose of harvesting carrageenan. And this is the same stuff we looked at in class that's used for a thickening agent and food like toothpaste and ice cream. And cultivated algae is often blighted by ice disease and epiphytic red algae. Ice disease is a wasting disease for algae. However, the focus is just epiphytic red algae. And epiphytic red algae is overlooked by farmers because it's not very well known. However, it is on the rise in the tropics, which is a big problem for them. Uh, Blighted algae is susceptible to damage like bacterial outbreaks, which results in decreased marketability. The reason why this happens is the attachments of the epiphytic algae leave holes or lesions within the host, which leaves a perfect opportunity for bacteria. Uh, the specific aim of this paper is to understand epiphytic dynamics and how environmental factors like temperature and salinity affect epiphytic outbreaks as well.
Alrighty, diving into our materials and methods, algae was ultimately collected from four Malaysian towns listed right below. In a laboratory setting, the algae ended up being fixed onto a 10% formalin. This helped with the preservation of the thallus by preventing its decomposition and decay. Following this, the epiphytes were removed and stained with the cotton blue solution. Next step after this was preparing for histological analysis. This basically entails that the algae was fixed onto a solution called glutaraldehyde, and in addition was also dehydrated in ethanol. Samples were also coated with xylene and injected with paraffin. All of this was done for the preparation of viewing it under a scanning electron microscope. On top of this, temperature and salinity was also taken 3 times a day from each respected site. Alrighty, going off our materials and methods, some of our results basically tell us that the dominant infecting epiphyte was Neosiphonia saviteri. We also derived that invasion of epiphytes is most prevalent in dry seasons. And we also concluded with that changes in seawater temperature and salinity share a positive relationship with epiphyte emergence. Some of the key takeaways from this is that seasonal patterns influence epiphytic algae prevalence. We also figured that environmental cues trigger epiphyte outbreaks, which is pretty self-explanatory. And we also want to be able to ensure that sustainable seaweed farming through the effective management of epiphytic growth. So going off my first point in determining the prominent epiphytic algae, Neosiphonia ended up being the most dominant compared to the other 4 algae species found on our focused algae. So here we can see that Neosiphonia is drastically outcompeting the 4 other ones. The following slides will go into more detail about the positive relationship of salinity, temperature, and what time frames these epiphytes are commonly most found in.
Okay, now we have imagery of the epiphytes on the thallus and some micrographs as well. So in figure 2, image A, we have what the epiphytes look like when they first attach to the host. And they look like just little black spores on the surface. And then in image B, we can see a tetrasporuling in between the inner and outer cortex of the host cells. And then now in C, we see what they look like when they're germlings and they penetrate through the epidermis of the host thallus. And then finally in D, we can see what they look like as adults on the thallus. And so here in figure 3, image A, we have scanning electron microscopy micrographs displaying the attachments of the epiphytes on the host. And then in image B, we have the actual lesions that these epiphytes leave behind that are caused by uh, their attachments. And these holes cause openings that let bacteria in and cause bacterial outbreaks. And that causes a decreased marketability. And now we have the histology analysis of the lab. And in image A, we can see a histology section of the branch of the epiphyte and the host thallus. Number 1 shows the thallus, or excuse me, the branch of the epiphyte. And then number 2 shows the rhizoid of the epiphyte penetrating the cortex of the host cell. And the host cell is marked as number 3. And then in image B, we can see an image that focuses more on the rhizoid of the epiphyte. Image 2 shows a rhizoid going into the inner cortex of the host cell. And then image 2 shows the outer cortex being penetrated by the rhizoid. Here we have a figure on salinity and temperature fluctuations and how they affect the epiphyte occurrences. So this is just following back to what Ivan was saying in his previous slides. Uh, Occurrences of epiphytes occur highest when there's high salinity and high temperature. And this happens during late February and early March. And then as time goes on, we can see that uh, at when salinity and temperature goes down, so do epiphyte occurrences.
So, looking at our outcomes, our results, and some of the information we were able to derived, it makes sense for algae and epiphytic species alike to be highly productive during warmer waters because it increases photosynthetic rates um, as it's demanded by its increasing metabolic rate. Um, and with this perpetuating substantial growth and development, uh, the paper doesn't go into depth about the increasing salt in conjunction with increasing temperature um, that would ultimately, as the paper states, push for a higher abundance of epiphytes. The only possible mention that the paper makes at this is by providing examples of these two abiotic factors as environmental cues for the occurrence of epiphytes, but it never goes into depth as to why physically you would have higher salinity in higher temperature waters.